Hello everybody. Today I will be talking about experiments and observational studies from part 3, and I will be doing chapter 13 part 2, which is more complex experiments. The first thing we need to talk about is blocking. When experimental groups are similar, we should block them. We do this so that we can isolate the variability due to the differences between all of the blocks, so that we can ultimately see the differences due to the treatment. When randomization occurs within the blocks, we call the design a randomized block design. Here we can see an example. Of course, we need to have random and randomization going on. So we take our volunteers and we split them between two different groups. Each group does get the same treatment, but then we want to compare so that we can see the differences between these two groups. So some things to remember about blocking is that it is the same idea for experiments as stratifying is for sampling. Like I said, we use blocks to reduce variability because we want to see the effects of the factors, not the blocks themselves. In this last example that we had here, we cared about the effects of our treatment, not males versus females, but we wanted to see the differences that males versus females had. Lastly, both methods group together similar subjects, and then we randomize them to remove unwanted variation. So there is a special type of blocking called matching. It's a special type of blocking designed for when subjects are paired because of similar traits not being studied. The big thing about blocking and blocking that includes matching is that we want to reduce unwanted variability. Here we have an example of the matched pair design, and we still randomize our treatment. And then we set up our normal blocking, but we have these two groups, and these two subjects. So they're all paired 2, 2, and 2. We have 18-year-olds, 27-year-olds, and 40-year-olds. We still care about what our treatment and the control is doing, and we're comparing our data, but we want to see 18-year-olds, 27, and 40. We do this again to get rid of unwanted variation, and even though that it's not being tested, we still match them by another similar trait. Another thing to talk about is factors. There's oftentimes multiple different factors that are included and examined whenever we look at these different combinations. So we still have a random assignment here, and for our example, we're looking at zucchini plants. Each group here contains 2 plants, and we have 6 groups. And then each group is getting a different combination though. That is the factor there. So here we see control, no water. Half a dose, no water. Full dose, no water. Then it flips and it goes control, water, half a dose, water, and full dose, water. At the end, we look and compare the end result. So shifting gears a little bit, we want to talk about what is statistically significant. At this case, obviously in our later chapters, we learned that sometimes there are specific values that we look at for what is statistically significant. But when we learned about Unit 3, we talked about the differences that are larger than what we would get from randomization is what we consider statistically significant. Now moving on to our 4 principles of experimental design, we have control, randomization, replication, and blocking. Our first principle is controlling. We want to be able to control the sources of variation to make the conditions similar for all treatment groups. This reduces variability, which is a huge part of blocking. Similar to control, randomization plays a huge part in experimental design. We want to equalize the effects of the unknown sources of variation. It doesn't completely eliminate these groups, but it does spread it across evenly to all treatment. Without randomization, our experiment is not valid. This is one of the most important things to keep in mind from part 3. So our third principle is replication. Repeating experiments by applying the same treatments to a number of subjects is replicating. We want to have validity in all of our experiments. So we want to make sure that we're getting the same results and not having any crazy outliers. Another thing to keep in mind is the outcome of one subject is an anecdote, not data. In order to get data, we have to repeat our experiment many times. Replication of an entire experiment when controlled with controlled sources of variation at different levels is an essential step in science. For our 4th principle, we have blocking. We group these similar subjects together and randomize them within each block so we were able to remove lots of the variability due to the differences between the blocks. Something to also keep in mind is that blocking isn't a required step, but it is an important compromise between control and randomization. The last thing to remember is that the best experiments are usually randomized, comparative, double-blind, and placebo-controlled. That is lesson 13.2, and now you will have your short answer. So here's a quick video of what the short answer review sheet for Unit 3, Chapter 13, Part 2 looks like. It is these 4 questions. I'm going to have the answers on the next page, and I will show it next. So go ahead and stop the video here to work on these, and then come back and check your answers. So here are our answers. So as you can see here, the first 3, I will just explain this down here. I asked you to design an experiment, but just a note, this is not a blocked experiment. I just wanted to have a review and there of what a regular designing an experiment is like. If you were to block this, you could potentially put in like boys and girls, or you could do a matched pairs design in here, and you could say junior girls, or you could say juniors, seniors, sophomores, freshmen. There's all sorts of ways that you can block designs, but here's the very, very basic way of doing a regular experimental design. And then here are your 3 other answers. If you have any questions, I'm sure you could ask our group and we could help you out. So that is Unit 3, Chapter 13, Part 2.